This is chapter 12, tumor immunology, immunotherapy. So we just want avoiding immune destruction is one of the hallmark of cancers. So for our body, we have three layers of immune defense. First is mechanical, so our like skin, our mucosal layer within our lungs, and like our guts. So that's like the first layer that's like physically preventing from like pathogens like bacteria coming into our body. And after that we have like a chemical defenses like complement systems or like lysozyme that as a complement system, like enzyme, they're like some sort of little proteins that like helps to break down or like attack those pathogens. And after that, if it doesn't work well, now our real immune system that kicks in with like innate, that's composed of innate immunity and adaptive immunity. So innate immunity it's like first kicks in and it's kind of more general purposes and it's just available everywhere, ready to go. That's how our body normally deals with like little pathogens. Like but if the innate immunity is not enough, then we get adaptive immunity kicks in and makes more specific strategy or ways to deal with the pathogens to that specific to the pathogens. And of course, in order to do that, it takes some time. So it doesn't happen every time, but it only happens after innate immunity is like struggling to do so. And innate immunity and like adaptive immunity kind of like interact with each other, so that like they kind of like boost each other's efficiency. So three ways that immune system is protecting against cancer: protect from cancer-causing pathogens like bacteria and viruses, great. Fight cancer causing inflammation, and recognize and kill tumor cells directly. We'll talk more into it. This is just a hematopoietic lineage, so our like little blood cells that also happens to be immune cells. A lot of immune cell stuff. So we have T cell, B cell, macrophage, granulocytes, dendritic cells. We'll go more into it. And this is in our lymph nodes. So it's just a system where like antigens can like come in, like this is hologen coming in, often with like the antigen presenting cells like dendritic cells. And dendritic comes in and it helps to mature T cells and B cells with those antigens. So they're specific to those, designed to or mature to deal with those antigens, and then goes to other parts of the body and deal with this. So this is part of the adaptive immune system. There are MHC classes 1 and 2 on how we deal with the pathogens. So MHC class 1 is presented in like is in every cells in our body. So it's basically when there's like viruses or other antigens like comes in, we present parts of it, those like antigens, or the intruders or the bad stuff to the surface of the cells. And if the surface of the cells has the CD8 plus T cells, which is known as cytotoxic T cells, they binds to that antigen here with the T cell receptor. And once it does, and if the T cell recognizes, oh, that this is not a great foreign molecules to have, then CD8 T cells, as the name suggests, so cytotoxic T cells kills this cell that presented this MHC, this antigen through the MHC class 1. So this can be used to recognize cancer cells and kill the cancer cells. So this is one of the mechanisms that our body removes cancer cells or tumor cell. And then the other narrowed way, MHC class 2. So this is what's happening is that there's exogenous antigens, and we kind of break it down, and then we present it on the surface of the cell with the MHC class 2, and then CD4 plus T cell comes in and binds with it with its own T cell receptor as well over here. And CD4 T cells is a helper T cell. So what it does is like basically make an alarm that, hey, we need more help here. So T cells is one of the antigen presenting. But ignore it. Anyway, so there's that. So antigen comes in, and the antigen presenting cells stimulate cytotoxic T cells, helper T cells. And cytotoxic like kills a bunch of stuff And the helper T cell activates B cells, and B cell can either make a plasma cell that makes like that secretes antibodies to help like deal with like the specific antigen that was like activated this in the first place. And also you can make some like memory B cells. So for the future, it stores some sorts of B cells that has a memory on how to deal with the antigen. So if the antigen comes in like a couple years later, we know we can kind of look through these memory B cells and then like find, okay, we have this memory B cell that as a that works against this antigen. Then we can just replicate from these memory B cells. We can like make plasma cells that produces antibodies that deals with this antigen. And blah blah blah. So cytotoxic T cells have main defense against cancer and pathogens. Great humoral versus cell mediated. So plasma cells makes antibodies, things that I explained earlier. Cytotoxics like kills target cells, yada yada yada. Antibody, it looks like that. There's a variable region at the end of it, so this is where the antigen binds. So that determines the affinity towards the antigen. And then there's in terms of structure, and then other parts is called just constant region, because it's just constant across different antibodies. Because what we really need to change is just this variable region that binds with or react with different antigens, but the main body itself can stay just as it is. So it's called constant region. And this main, like this too long like thingy, is called it's a heavy chain, and this like smaller shorter one is called light chain. But there's that. And so the important part is this antigen binding site in this variable region, right? So, and it has like a bunch of genes that like makes this composes the variable region. So we have this V for variable, C for constant, and then the D and J, which stand for diversity and joint or something. But anyway, the point is that this V, D, J are all contributing factor into a variable region, and with the various and they randomly shuffles this gene to make this VDJ complex to make the variable region. So if we have this 10 J segments, 200 V segments, and 10 D segments, and we can, and with a bunch of other stuff, like we can make like 2 million like combination, and we after it's made, we test them with our body and see if it reacts. Because it's randomly made, we don't know how it's going to work. So we test it all against ourselves. If it does react with itself, then it shouldn't be used, because the antibody is basically attacking or helping to attack what is binding to, right? So we don't want to attack our body, and that will cause autoimmune disease. So this VDJ, and then we test whether, and if it does not like react with our body, this newly made antibody, then we can store them and then use them to deal with outside pathogen. Okay, so cancer immunity cycle. So T cells like lyses these tumor cells. This is an antigen receptor, and then it's gonna like release a bunch of like antigen within the tumor cell, right? Because we stabbed the tumor cells and killed it. And then so that released, this death of tumor cell leads to the release of cancer antigen, and that antigen is picked up by this APC, the antigen presenting cells like dendritic cells and a macrophage. A macrophage? I don't know. Actually, let's look it up. Yeah, it was a macrophage. So macrophage, dendritic cell, and B cells. Anyway, so those three are the main guys: B cell, dendritic, and macrophage. So anyway, those guys have the MHC2, which is what we talked about over here. So it's something that like brings help. It's like sending an alarm. So and then so this APC handed off this antigen to this T cell, and T cell receives it with this T cell receptor. And okay, so this APC tells, hey, kill this guy, kill the guys with this type of antigen, and T cell, okay, I'm gonna kill them. And then they get transported into a tumor site, and then the T cells like kill the tumor cells, and the tumor cell death leads to more release of cancer antigen and APCs and T cell interaction again, and then it goes on and so forth. So it's a positive cycle, great. So there are two different types of antigen. There's a new antigen, means like new antigen, right? So it's a new antigen that is only specific to tumors, that has occurred because of the mutation that's caused by tumor or caused within the tumor. And the tumor-associated antigen is just an antigen that's kind of associated with tumor. So neoantigen works really well because it's specific to tumor, but it's not always available. And tumor-associated antigen is more like a broadly applicable. So we try tumor-associated antigen first. And the role of the innate immune in tumor suppression, there's an NK cell which stands for natural killer cells, and they're like somewhere in between like innate and adaptive immune cells. It's kind of weird, but anyway, they're kind of counted for both. But anyway, so natural killer cell as a first line of defense, when they have this like a stress ligand receptor like this, and then they bind, and then there's like MH1 like one that's presented here, and then they're like kill the cells, like the cytotoxic T cells that we talked about here, up here, like here, right? So CD8 T cells like kill these cells that present these weird molecules from outside. So it's not only the CD8 T cells, but it's also NK cells. So that's great. And and then there's these tumor cells here. So it's been laying around for a long time, and the first line of defense, the innate immune system kind of deals with it and produce an antibody specific to this. Then the FC, the antibodies bind with this tumor cells, and then the NK cells receptor, FC receptor binds with it as well, and then allows the antibodies, NK cells to kill these tumor cells. And there's just macrophage with the antibodies on it, and then it just kills the tumor cells. And what's happening here with these tumor cells is that, so there's a DNA in the cytoplasm, so which is not really where the DNA is supposed to be. It can indicate that there's a virus that injected with this RNA into the cells, and then the RNA turned into DNA. It could be virus DNA, which is bad for the cells. So it's going to do something like go through like sting, whatever that may be, and then it's going to release the interference. It's basically like screaming, and they're saying like, I have a bad DNA in myself, and I need to be killed, and that will cause to bring more like other cells in these immune cells to be recruited and activated. So there's that. Immunity checkpoints and immunotherapy. So if the cancer cells can bypass this immunity checkpoint, then they can survive. So CTLA, and they got Nobel Prize for it. Great. Antigen presenting cells again here with interacting with T cells with MHC complex and the TCR. With the antigen in here. So that's like the first step activation, but we also want to like make sure that like it's getting properly activated. So it's not just randomly flowing antigen, it just happens to, like get bind to the TCR of the T cells, and then we don't want T cells to start making like different, like get activated and start killing anything that's related to AG, like, and kill basically destruct our own body. So we have this like a other structure here that kind of co-stimulated, so called B7 on the APC and the CD28 receptors on the T cells. So what the B7 CD28 are, they're making sure they're getting properly activated. So this is another one of the checkpoint. Another thing, so if there's one thing that kind of stimulates, we have to have something that inhibits for the control, right? So we have the B7 on the APC, but on the T cell, it also has CTLA4, which is like a co-inhibitor. So it competes with the CD28 to slow down the stuff. So when there's CTLA4 is like next to TCR, and then it may, instead of this B7 getting bind to CD28, it may bind to the CTLA4, and hence like doesn't activate the T cells. So great. So this is inhibiting T cells from killing stuff. So it can be one of the things that can be exploited by the cancer cells to evade the death by T cells. Tumor cells also evade the T cell by the PD-L1, because what that does is it binds with like T cells, and then it suppresses the T cell cytotoxic response. So PD-L1, the more the tumor have, they're more likely to survive. So for the immune checkpoint inhibitors, there's different drugs to kind of deal with this. So this whatever drug it called, ipilimumab, whatever that thing is, it binds with the CTLA4, which is like inhibitory. So if you already just like bind with it, so it doesn't activate the T cell well, so then B7 will bind with CD28 instead, and then T cell will get activated and kill more stuff. But for tumor with PD-L1 and PD-1 interaction, we can kind of like prevent their interaction with different drugs on each stuff. So kill. But this kind of, so in theory, I feel like it should work well with like golden bullets, and it kills all the tumors. But it doesn't really work well, because if cancer cells already learn to evade the immune cells so that this interaction doesn't happen at all, which means like T cell is not able to recognize tumors, hence this interaction happens anyway, then even if you put these molecules in the patient's body, it's going to do nothing. That's why some respond well to this kind of treatment, some doesn't. Depends on the stage where the cancers are at. But we're going to talk more into the coming slides. So it's just a different stuff. So this is control, and then this is the drug. And with drug, the red and purple, it has a better survival rate. The y-axis is the survival rate, and then this is like years later treatment. So great. It's still 25%, which is better than 0%. So better than nothing. So immunoediting is the host immune system shapes tumor immunogenicity, and cancer cell modulate and edit the host anti-tumor immune system. The immune cells are killing cells, but it also pushes the cancer cell in the process to evolve to survive, right? Because it's killing all the cells, cancer cells that get detected, but the one that gets undetected will survive and proliferate, and the more there are going to be more cancer cells that's going to survive later on, right? So there are three phases of this immunoediting. One is the elimination of the cancer, but that's great if it happens, if it gets eliminated. But equilibrium, the selection of less immunogenic tumor cells during an anti-tumor response. But in the process of tumor cells, getting like recognized, like so that the immune cells they try to find the tumor cells and kill them, but in the process we miss some tumor cells that are more invisible or less likely to get recognized, and they proliferate. So that's what has happened, the selection of less immunogenic, less recognizable tumor cells. And then on the third phase, escape whereby the tumor evades the immune system, so now they are invisible to the immune system. So evidence, the tumor formed in the immunodeficient system are more immunogenic, in other words, more visible than tumor formed in immunocompetent host. So when there's no immune system in the mice and there's tumor, they don't have to evade the immune system. So when you get the tumor, those cancer cell from the immunodeficient mice and put it somewhere else, they're more recognizable than the tumor that's developed in the immunocompetent host. And the profile of tumor infiltrating lymphocyte, which is just this lymphocyte that goes into tumor and fight, correlates with cancer patient survival and prognosis. So it depends on the type of the cells, the immune cell that goes into the tumor and fight. They correlate with the cancer patient survival rate, because it makes sense that the better army you send in to fight, the better survival rate you have. And also it could be used for prognosis, the immunoscore in other words, and it apparently works well for cancer diagnosis. Mechanism of tumor evasion, so less of tumor antigen, so if there's no antigen, you can't respond to it. Downregulation of MHCs is basically saying like, you're not showing your stuff. So MHC is something that's like coming from the sick cell itself here. So this is presented by and this is MHC. So if you don't have an MHC, you don't have a way to kind of show to other like innate cells, right? The immune cells, right? So it won't work. So there's another one, downregulation of MHC, and resistance to cytotoxic pathway, overexpression of immune checkpoint proteins. So even if you get detected by the immune cells, like you can kind of like do the thing like this, CTLA4 or this kind of thing that kind of suppresses the T cells killing the cytotoxicity. So it's like, yeah, like you got me already, you see me, but don't kill me. That's what's happening. Expression of immunosuppressive molecules, immunosuppression. So no immune system work as hard. Vaccine. So vaccine is composed of antigen and adjuvant. Adjuvant is something like a molecule that kind of like activates the immune system. So and like help antigen to get processed. So there's that. So it's kind of like hey, look what we have, this antigen. So what's happening is that we get this antigen from through biopsy of the patients, right? And then we get the antigen from them. And we inject this antigen, and then they get acquired by dendritic cells or other antigen presenting cells, and they get processed, and then they get presented to T cells, MHC plus antigen presenting cell, which happens in the lymphoid organ, which we talked about at the very beginning over here in the lymphoid. There's T cell coming in with the antigen, and it reacts with T cell and B cell, and it's like we make B cells and T cells specific to those antigen. So and then so the cytotoxic T cells goes to the tumor, and then they just like kill the tumors and tumor cell death. That's great. So this therapeutic cancer vaccine targets neoantigen, which is the new cancer specific antigen, right? But so ideally the antigen needs to be tumor specific, expressed in every tumor and critical for survival of the tumor. But that's not always the case. This is an ideal case. So if it's not tumor specific, it can kill other cells and cause side effect expressed in every tumor. Well, if it's not the case, then it's going to miss some of the tumors and it's going to survive. And critical for the survival of the tumor. So if the antigen is not critical for the survival of tumors, even if T cell kind of deals with the antigen, like it may not kill the tumor. So great. So vaccines are great if everything's work out, but it's not always the case. So vaccines are much more effective by stimulating the immune system rather than targeting the tumor, because we're targeting the immune cells, right? We're not directly killing the tumors with it. Regular vaccines are preventative. Most cancer vaccines are tumor fighting, so the flu vaccines, they're like preventative measure, but cancer vaccines are after we get infected by it, other than HPV. So it's a little different. It doesn't work that well because for the reason, the ultimate goal is that we get the preventative vaccines for like people who are susceptible to the type of cancer. But it's not always working that well because the immune system gets weaker as we age or we have experienced chemotherapy. Because chemotherapy kills these cancer cells, and cancer cells are in order to target cancer cells, we target cells that are rapidly dividing, including our skins or blood cells, which also include immun cells. So we already kill a bunch of the blood cells and immune cells. So even if you try the immune cells immunotherapy after we go through chemotherapy, it's not going to work as well. And so resistance may arise as usual for all cancer therapies. They're more effective in early stage of cancers, but you know, like it's difficult to have a trial because it's like costs a lot of money, and like most people just go through regular like chemical drugs that kills like cancers rather than immunotherapy. And it's difficult to generate mouse model because mouse models are not going to work the same as the human immune systems. So that's that. On the last slide, this is this thing called CAR, which stands for chimeric antigen receptor. Basically, it's a combination of the antibody and the T cell receptor. So T cell receptor is in T cell, right? And that's how T cell get activated, like with this kind of thing. So what they did is there's a TCR, but then there's instead of antigen, we put like antibody here on it instead. So when the T cell, so whenever the antibody is, let me draw. So we have this thing, basically. This is antibody, and this antibody reacts to this cancer, right? And then so this gets active, then binding with cancer cells, activates antibody binds with activate T cell receptors, so T cells are activated, and T cells start to kill the cancer cells. Great, it works actually quite good. So how it works is you get the T cells out of the patient's blood, and then you put this CAR, the chimeric antigen receptor gene, into the T cell, so what you're doing, so that T cells can express this CAR stuff on their surface naturally. So and we make a bunch of these T cells injected back into to the patient, and then those CAR T cells will like target the cancer cells. But again, it's really expensive and it goes through a lot of stuff, so it's not widely used.